Hello and welcome to the program of second year Bachelor of Science, subject Botany, semester 4. Course code is BOC 104 and the course title is Plant Physiology. Title of the unit, Unit 4 Enzymes. Module name, Enzyme Inhibition, Part 1. In this module, I'll be defining enzyme. I'll be talking about the different factors affecting activity of enzymes. Then I'll be defining the enzyme inhibitor, explaining enzyme inhibition, types of enzyme inhibition and types of reversible inhibition. At the end of this e-module, student defines enzyme inhibitor, explains types of inhibition, defines reversible inhibition, explains types of reversible inhibition and differentiates between competitive and non-competitive inhibition. What is an enzyme? It's a biocatalyst that accelerates the rate of reaction in a cell. Basically, enzyme is a protein molecule that always increases the rate of reaction in a cell. So, enzyme is called as a biocatalyst. There are a number of factors that affect the enzyme activity, and these factors are substrate concentration, enzyme concentration, temperature, pH, ionic strength and enzyme inhibitors. Now coming to enzyme inhibitor. What are enzyme inhibitor? Molecule that binds with an enzyme and brings about a decrease in catalytic activity of that enzyme is called as enzyme inhibitor. Then what is enzyme inhibition? Enzyme inhibition is a process in which the inhibitor binds with an enzyme to decrease or to stop the enzyme activity. Enzyme inhibition is further divided into three types: reversible inhibition, irreversible inhibition and allosteric inhibition. Reversible inhibition. In this type of inhibition, the inhibitor binds non-covalently with the enzyme and the enzyme inhibition can be reversed. It can be reversed because of two things. Number one, the inhibitor binds with an enzyme with a weak bond, and that's why the inhibitor can be separated by dialysis. Secondly, this inhibitor, it cannot function properly if the substrate concentration is increased. So, when the substrate concentration is increased, the number of substrate molecules will increase and therefore they will bind with the active site and bring about the reversible reaction. Reversible inhibition is further divided into competitive, non-competitive and uncompetitive inhibition. Now coming to the competitive inhibition. What is this competitive inhibition? In this type of inhibition, the inhibitor shows the structural similarity with the substrate. As the inhibitor shows the structural similarity with the substrate, both of them compete for the active site of the enzyme. Inhibitor as well as substrate, both of them are going to compete to bind to the active site of the enzyme. If the inhibitor succeeds, it will bind to the active site and it will block the active site. As a result, the substrate no longer binds to the active site. It does not get a place to bind to the active site and thus the substrate remains unused. And so we say that the inhibitor has inhibited the enzyme activity. The very best example of competitive inhibition is the succinate that acts as a substrate. And this succinate, it binds with succinic dehydrogenase enzyme to produce a product or to give a product called as fumarate. There is an inhibitor called malonate. The structure of malonate is similar to that of succinate. So, there is a competition between malonate and succinate. If malonate succeeds, it goes and binds with the succinic dehydrogenase, blocks the active site and doesn't allow the succinate to form the enzyme substrate complex. As a result, succinate remains unused and does not form fumarate. So this is a very good example of competitive inhibition where both inhibitor and the substrate are competing. Coming to the non-competitive inhibition, in this there is no similarity between the structure of inhibitor and the substrate. But the inhibitor goes and binds to the enzyme, not to the active site but to the other site on the enzyme. As a result, the enzyme inhibitor complex is formed. The inhibitor, it will change the structure of the enzyme. It will bring about the conformational change. As a result, substrate cannot bind to the active site of the enzyme and as such, the substrate remains unused, the enzyme remains non-functional. And such a type of inhibition is called as non-competitive inhibition. Coming to uncompetitive inhibition, in this type of inhibition, there is an enzyme molecule, inhibitor and the substrate molecule. First, the substrate will bind to the active site of an enzyme to form an enzyme substrate complex. Now, the inhibitor will bind to the enzyme substrate complex. Here what is important is inhibitor alone cannot bind to the enzyme, but it will bind only to the enzyme substrate complex and then it is going to make the enzyme non-functional, inactive, so the catalytic reaction does not take place. So, in all the three types of inhibition of a reversible inhibition, the first is competitive, second is the non-competitive and the third is uncompetitive inhibition. These are the references for this e-module. Thank you.